Hello, everyone. My name is Lei Tian. I'm a PhD student working with Dr. Hosami Dost at McMaster University. In this symposium, we have heard a lot of awesome direct antimicrobial applications of phages. So I would like to bring some biomaterial elements to the discussion. Today, I will introduce our work on phage hydrogels. First of all, what is a hydrogel? It is usually a network of cross-linked polymers chains that can absorb large quantity of water without dissolving. So hydrogels, they are soft and highly hydrated, like human tissues. And because of these properties, scientists have found many applications on it. For example, tissue engineering, diapers, drug delivery, last but not least, wood dressing and contact lenses. So hydrogels are mostly made of polymers, but in fact, we have other options in our research. Our hydrogels are made of bacteriophages. We work with a specific kind of bacteriophage called M13 phage. You can see here is a schematic of M13 phage with its 5 structural proteins along with a diagram of the M13 genome with only 11 genes. You can see the phages are filamentous. They are like nanofibers. So how do we cross-link these nanofibers to form a network? An easy solution is chemical cross-linker. For cross-linking, we can target various functional groups on the M13 capsid or like coating or the coat. However, it has been shown that the lysine residue here, like the are much more efficient targets for chemical reactions. A single M13 phage have 5,400 available lysine residues. We chose a very common cross-linker glutaraldehyde. You can see the equation of cross-linking reaction here. Glutaraldehyde is the aggressive carbonyl reagent that condenses amine via manic reaction and conductive amination. It was commonly used to prepare antibody enzyme conjugates. Glutaraldehyde react with lysine residues on the phage coat protein. They can react at room temperature and near neutral pH. We firstly investigate the concentration range of phage that can form a hydrogel. When we keep the glutaraldehyde concentration as 1% consistently, any phage concentration lower than 10 to the 13 PFU per ml took more than 1 week to gel and end up to be very soft. So for the rest of the experiment, we focus on the concentrations higher than 10 to the 13. We found out our phage hydrogel can absorb a lot of water. You can see here that M13 hydrogels can absorb up to 16 times their weight in water. The figure tells us the hydrogel made with lower phage and cross-linker concentration can absorb more water. We think this trend is because of the change of the pore structure. So we check the micro and nanostructure of the phage gels made at different concentrations. You can see here the hydrogel made with less phages have larger pores, giving stronger absorbing ability. And you can see the filaments with size suggests the M13 filaments bound of several hundred phages. A very interesting property we observed was autofluorescence. The phage solution and glutaraldehyde themselves are not fluorescent, but the hydrogels made of these two components showed autofluorescence at different excitation wavelengths. We hypothesize this property is the result of the cross-linking reaction. The UV spectrum showed a distinct peaks for the hydrogels, which is not present of phage solution. The FTIR peaks show two fingerprint peaks corresponding to amide. There are multiple new absorption peaks. The most important one is imine. The carbon-nitrogen double bond, which can be the reason of fluorescent property. Because phages are mostly proteins, we hypothesize phage hydrogels are biodegradable. To prove that, we put a hydrogel in protein SK solution. After 36 hours, the gel was mostly digested, and you can see the color change in the degradation product that shows autofluorescent property, meaning the release of the fluorescent molecules produced in the cross-linking process. Another interesting property that phage hydrogels can hear. Here you can see two hydrogels have been cut and were in contact with each other in calcium. After 34 hours, it can heal itself and regenerate. We also did a mechanical test for the self-healed hydrogel, although they didn't show a mechanical strength as much as original gel. They have a reasonable strength after healing. To test the bioactivity of phage hydrogels, we incubate hydrogels with E. coli, which is the host bacterium of M13. The final solution showed a high concentration of phage, indicating the M13 inside the hydrogel are still infective. On the contrary, the hydrogel incubated with pure LB has no phage, which proves that M13 inside the hydrogel didn't leak out. Instead, they infect E. coli in situ. There are mainly 3 to 4 other labs worldwide are working on pure phage hydrogels. We are the first lab proposing that phage hydrogels have the properties of self-healing, hierarchical structure, fluorescence, and bioactivity. So what can these gels be used other than antimicrobial application? We can easily engineer the genome to express different peptides on the phage coat surface, especially G8 protein, which has 2,700 copies. If the phage express a peptide that like to bind to cancer cells, this hydrogel can be used for cancer screening. Lastly, I would like to thank my team. This work was started by Asday, who was a master student in our lab. After she graduated, I'm leading a team to further investigate the application of phage hydrogels and phage microgels. This work has been published and covered feature on chemistry of materials. We are honored that some social medias are interested in this work and reported it. If you're interested, please visit our website and watch an interview of my supervisor, Dr. Hosseini Dost from CBC, the National. That's all. Thank you.